Hello, I'm Joelle, a member of the Waldron Group at MSU, and today I'm going to be talking about a recent paper by Teresa Hwang from the Keating lab at MIT. So just a little vocabulary first. Short linear motifs, or SLIMs, are just 4 to 6 residue long sequences that are critical to protein-protein interaction. And while those residues within the actual motifs have been decently studied, it appears that the surrounding sequence and its contextual implications on that sequence recognition are not very well understood. So that's what they aim to do. ENA is the protein that they studied. It's an actin regulator, and they chose this because it's upregulated in some particularly invasive cancers. The domain EDH1 is involved in recognizing some extended SLIMs. So extended SLIMs just being those that aren't 4 to 6 residues long. They may be longer, but they contain 4 to 6 residues that are definitive of the motif itself. So the particular SLIM that they looked at is called FP4. You can see the sequence right down here. And so X being any residue really could be multiple residues, which is what they mean by extended SLIM. And so they went and looked at the human proteome to see how many instances of FP4 there were. And there were nearly 5,000 occurrences of just different FP4 variants. So from that, they kind of said, well, sequence context has to matter if there are so many instances of these. There's no way that surrounding sequences don't affect the recognition, right? And so they devised this scheme called mass titer to further inspect that hypothesis. They use a bacterial surface display as shown in figure A, as well as fluorescence activated cell sorting, just an example plot shown here in figure A. And then this mass titer scheme is really well explained in figure B. Basically, they performed a FACS sort into 4 affinity gates, sorting different FP4 sequences. And they did this at 3 different concentrations of ENA. So once they collected all of those binders, they analyzed how many of them, out of the 108 unique binders that they collected, they analyzed how many were canonical FP4 sequences, which, if you're curious, is shown down here. They also provided frequency plots for sequences that were either in the human proteome, in their input library, or that were mass titer hits. And from all of this, it's very impressive. They were able to identify 19 previously unreported interactions between ENA. And they actually indicated the subcellular location. Those in white have been previously reported, but you can see that they reported quite a few novel ones. So this method seems to be a good one. But if you're interested, I highly recommend this paper.